Hi, my name is Ben Covert, and I wanted to share my amazing summer research experience through the USDA Circularity and Digitization REEU. Today, I'd like to speak about the project, its goals, and what I've learned, and how I can continue to use these skills in the coming semesters here at UF. Climate change poses an ever-increasing threat to our world and must be responded to in order to ensure a food-secure future. Rising sea levels have been shown to increase rates of saltwater intrusion into soil. This can lead to stunted growth, physiological changes, and crop death over time. To adapt to these environmental changes, new potentially salt-tolerant cultivars are being researched across the country. Over the past 10 weeks, I have had a few main goals governing my work. The first main objective was to connect a collection of sensors in a controlled growth chamber to a virtual machine. This virtual machine would serve as an interface between the sensors and cloud computing services like InfluxDB and Grafana. This proved to be more challenging than expected because of the connectivity issues in the chamber. However, an improvised solution was found where data was recorded to a computer in the chamber and later uploaded to the cloud. The second objective involved a sampling of plants in the growth chamber for leaf position, angle, and length. This data will be used to create a crop model for growth patterns. After learning about current research being conducted in the Agricultural and Biological Engineering Department, I moved on to the question, what sensors would be necessary to phenotype different Brassicaceae? And I came to the conclusion that there are some good contenders. Atlas sensors, such as their pH, dissolved oxygen, electrical conductivity, and oxidation reduction potential sensors can be easily isolated to give accurate and precise data about water quality in a hydroponic environment or in a nutrient solution tank. A capacitive soil moisture sensor would be necessary in a high throughput, non-hydroponic setting to ensure the proper level of irrigation. RGB cameras could be used to automatically track the growth of the plants and compare the models with non-stressed controls. Finally, some CO2 sensors, such as an SCD30, would be necessary to track CO2, temperature, and humidity, and account for any anomalies. Moving forward, I could inform the design of a sensor suite that would monitor plant growth under stressed and unstressed conditions. The more automation is involved in this process, the faster the arduous process of phenotyping can occur. To make and test the sensor suite described above, I would need to plan, with more background and computational knowledge, design the apparatus and select specific sensors, test the module in a small-scale setting, and then launch and distribute the models to research partners engaged in the phenotyping process. One area that specifically requires attention is experimental location. The growth chambers I talked about earlier are heavily insulated and very challenging to pass any sort of wireless connection through. Another area of focus is, could less sensors be used if they were mobilized, thus cutting down on costs? Finally, is there any way to use a high-powered microcontroller, like a Raspberry Pi, to process images before they even get to a human's desk? Additionally, I've learned that some areas I would like to focus on during my last semesters are cloud computing solutions involving automation and also other methods of wireless communication, such as cellular or radio. Some of the skills I've gained this summer include proficiency coding with microcontrollers. Also, I learned about the many communication protocols that are used to control sensors and other devices. Examples of these include I2C, SPI, and UART. I also had the opportunity to spend a great deal of time working with and operating the hyperspectral camera in the ABE building. This camera can see beyond the human eye and notice patterns otherwise undetectable. This summer, I've learned about plant physiology, integrated circuits, programming, teamwork, problem solving, and scientific communication. It was an absolutely amazing experience, and I can't wait to see where my next steps in this journey take me. I'd like to give a big thank you to SURF, and I'd also like to thank this project's PI, Dr. Melanie Carell, for being so kind and willing to take time to support my academic growth. I'd also like to thank Drs. Boz, Martin, and Roberts for teaching our cohort so much about circularity, digitization, and the circular economy. Lastly, I would like to thank my coworkers, Paco and Kyle, for the everyday insight that helped me to succeed this summer. Thank you.